We're going to look at reproduction and specifically the two main types of getting a cell to make another cell. And we have asexual, which means there is one parent. The offspring are genetically identical to that one parent who's contributing the DNA. This means there's less genetic diversity since they're identical. And the positive is you can have a lot of offspring in a very short amount of time. So we're going to focus on the asexual, one parent contributing the DNA first. And then it will separate into two identical daughter cells. And cells that do have a nucleus, it's a little bit more complicated. And so we're going to go through something called the cell cycle. And so we can look at it in terms of just the different phases first. And there's two main phases, interphase and cell division. It spends over 3/4 of its life in this phase. And this phase is made up of three subphases: G1, which is the first growth phase; S phase, where DNA makes a copy of itself; and G2, where it basically gets ready for cell division. And during this time, you see cells that are in interphase. You can see the nucleolus, you can see the nuclear envelope, and the DNA looks kind of like a big blob. It's not really into distinct chromosomes. The second part, cell division, is divided into two different subgroups: mitosis, division of the nucleus, and cytokinesis, which is division of the cytoplasm and the cell itself. And so you can see the kind of the big picture. We start with one cell, we end up with two identical daughter cells. And so mitosis can be broken down further into PMAT. And you can remember that P stands for prophase, and the pairs are condensing. You start to see the distinct bundles or chromosomes. Metaphase, the chromosomes are moving to the middle. Remember that the centromere finally divides and two sister chromatids move to opposite ends. So we say they're moving away from the center in anaphase. And in telophase, the DNA is at the two ends, and you see the nuclear envelope starting to reform. We can look at that in a slightly different way, more like in a cycle, which is what is actually happening. So here is our one cell going in. Remember, I told you that it takes, it's probably in this first phase, interphase, about 3/4 of the time. So here's G1, S, and G2. The second part, which is called cell division, is divided into two parts: mitosis and cytokinesis. It's usually in this phase about 10 to 25% of the time, which is what you hopefully saw in the onion root cell lab. And then mitosis is actually divided into these four subphases: PMAT, prophase, metaphase, anaphase, telophase, which is PMAT. And as a result of this, we end up with two identical daughter cells. If we quickly look at what's happening during the cell cycle in eukaryotic cells, here is our DNA, and this is why it looks kind of like a big ball of unwound yarn. During prophase, it's going to condense into little bundles. So our chromosomes form like this. The reason the chromosome looks like an X is because it's actually made a copy of itself during S phase. And the last place it's attached is the centromere. And we call these two identical copies the sister chromatids. So this is just showing you another way of looking at what we just talked about. Once we get to division of the nucleus, mitosis, remember we start out with prophase. The pairs are condensing, so we see these distinct bundles. They start out diploid. That means they have two copies of DNA, one from our mom, one from our dad. During metaphase, they line up in the middle as the spindle fibers attach to the centromere. During anaphase, they move away from the center because the centromere breaks and each sister chromatid moves to either side. Telophase, notice that